Assalamu alaikum, myself Shoaib Ihlaq, and I'm going to present the topic sandwich ELISA test. Sandwich ELISA is a type of enzyme-linked amino-sorbent assay that measures the amount of antigen between two layers of antibodies, a capture antibody and a detection antibody. It is called a sandwich because your antigen is bound between two antibodies. Secondary enzyme-linked antibodies are also used in indirect type of sandwich ELISA. We can see in the picture that a round green color antigen is bound between a capture antibody and a detection antibody that is green in color. Secondary enzyme-linked antibody is also attached with the detection antibody. Principle: the sandwich ELISA test works on the principle that the surface of the well of microtiter plate is applied with a well-known amount of the capture antibodies. The antigen containing sample to the each well is added. A specific detection antibody against our antigen added, forming a sandwich, and add an enzyme-linked secondary antibody that binds specifically to non-specific region Fc of detection antibody. Then the substrate is added that is converted by the enzyme to a colorful product. Diagram. Let's look at the diagram. In A, well coated with capture antibodies and blocking solution. In B, mixed sample are added that bind to the capture antibodies. In C, detection antibodies are added that attach with the antigen. In D, enzyme-linked secondary antibodies are added that link with detection antibodies. After that, in E diagram, the substrate solution is added that is converted to a colorful product that is red in color. Let's look at the equipment and the reagent used in the sandwich ELISA test. Equipment that are used are microtiter plate, multi-channel pipettes, washer and reader. Reagent that are used are capture antibodies, blocking buffer, phosphate buffer saline, sample antigen, detection antibodies, enzyme-linked secondary antibodies, and substrate solution. General procedure of sandwich ELISA test. It has 3 steps. The first step is coating with capture antibodies. Coat the wells of PVC microtiter plate with the capture antibodies at a concentration of 1 to 10 microgram per milliliter in carbonate or bicarbonate buffer at pH of 9.6. Cover the plate with an adhesive plastic and incubate overnight at 4 degrees centigrade. Then remove the coating solution by washing twice with 200 microliter PPS solution that will remove unwanted or unbound capture antibodies. We can see in the video that capture antibodies are added that bind to the surface of the well. Then ionizing is done that removes the unwanted or unbound capture antibodies. The step 2 is blocking and adding samples. Block the remaining protein binding sites by adding 200 microliter blocking buffer per well. Then cover the plate with an adhesive plastic and incubate for at least 1 to 2 hours at room temperature or 4 degrees centigrade overnight. After that, add sample 100 microliter of appropriately diluted samples to the each well is added. We can see in the video that sample that a bacterium or antigen are added in the microtiter plate well that binds to the capture antibody. The step 3 is incubation with detection and secondary antibodies. Add detection antibodies 100 microliter diluted to the each well, then cover the plate with adhesive plastic 2 hours at room temperature. Add secondary antibodies that are conjugated with HRP or ALP enzymes 100 microliter in amount. Cover the plate 2 hours with adhesive plastic. Then wash the plate 2 4 times with PBS solution. Then add the substrate solution. 100 microliter of chromogen substrate solution is added that converts into the colorful product. We can see in the video that detection antibodies or enzyme linked scanty antibodies are added in indirect type of sandwich ELISA that binds to the antigen. In video 2 we can see that the substrate is added. The substrate changes to the colorful product that is blue in color. Let's look at the results. If the test is the positive, the color of the substrate solution changes. If the test is negative, there will be no change in the color of substrate solution. For example, TMB substrate changes from colorless to blue color by the action of HRP enzyme. We can see in the video, if we add TMP substrate in the solution, they will be changed into the blue color by the action of HRP. It means that the test is positive. We discuss about the introduction, we discuss about the principle and the procedure and the result of the sandwich. These are the contributors. Thanks for watching.